Hello friends, welcome to my channel STEM's Biology. Today we are going to discuss a very important topic. Can you guess from the board? Yes? From these three words, I think you may guess the topic. It is about genetics. So we are going to study about genetics today. And as we have these three scientists, and we know that these three scientists are the inventor, rediscoverer of the Mendel's work. So in the year 1900, they rediscovered the work of Gregor Johann Mendel. So we are going to study about the experiments done by the father of genetics, Gregor Johann Mendel. Gregor Johann Mendel. So he is known very well to us as the father of genetics, and the reason is that he is the only successful scientist in his era to give the detail about factor that we know as gene these days. So how genes are responsible in carrying the parental characters to their offsprings is described successfully by Gregor Johann Mendel. So look at this part of the board. Here I have the name of some scientists who worked on genetics before Gregor Johann Mendel. So they worked during pre-Mendelian era, and J. Kölreuter first worked on tobacco plant and found that the parental character transfers to the offsprings. The offsprings may be similar to either of the parents or intermediate between the two parents. But all these scientists, along with Buckner, Nordin and Gauss, all these scientists failed to explain, none of them explained with sure degree of repeatability, means they could not explain clearly how the characters of parents is going to be found in their offsprings. So how they are repeated in their offspring is not explained by any of them clearly. So Gregor Johann Mendel, and he worked on the discovery of genetics or different ideas. He proposed different theories and gave different laws of genetics. During the 19th century, he worked in the 19th century. Okay, so in the 19th century, between 1822 to 84, his birth at 1822 and he died in 84, and between that period he worked a lot on genetics. So he took the garden pea or the Pisum sativum. Before his works on genetics, Mendel knew nothing about genes and chromosome. He has no idea about genes and chromosome. So let's study about the work of Gregor Johann Mendel. We know Hugo de Vries, Erich Sermach and Karl Korens together rediscovered the work done by Gregor Johann Mendel, father of genetics, in the year 1900. So these three scientists rediscovered the work of Gregor Johann Mendel independently. Okay. So in the year between the year 1822 to 1884, Gregor Johann Mendel gave his greatest contribution towards the world of genetics. So why do Gregor Johann Mendel choose pea plant or the garden pea? Garden pea. Garden pea, the botanical name is well known to us, Pisum sativum. Pisum sativum is the botanical name of the pea plant. And why do Gregor Johann Mendel choose Pisum sativum for his experiments? Let's discuss about that. So the Pisum sativum has certain specific characters. That is the reason why he chose the pea plant or the garden pea. So what are the characters? Number one is controlled pollination. Controlled pollination. It can be pollinated in a controlled way. The second one could be easy to cultivate. Next one is easy to cultivate. It is very easy to cultivate the garden pea in the field. That is the reason why he chose the pea plant. Another character is that it has sharp characters. Means the pea plant shows sharp characters. Means the characters are very clearly seen. Next one is that it is a bisexual flower. The Pygium sativum is bisexual. It is bisexual. So both the male and female reproductive parts present in a single flower. Next one is it can be easily hybridized. Easy hybridization. Easy hybridization. So it can be hybridized easily, the Pygium sativum. Next one is that it can grow to maturity in a season. So it is annual plant and it can be grown to maturity. Can be grown in a season. In a single season, it can be grown to maturity. So these are the various important characters of the plant. Why Gregor Johann Mendel chose garden pea or the Pygium sativum. Controlled pollination, easy to cultivate, sharp characters, grown in a season or annual plant, bisexual flower, and easy to hybridize. So these are the very important thing that is present in a garden pea. So remember the chromosome number of garden pea is 14. It may be asked in the competitive examination. Next, self-fertilization can be prevented by removing anthers through the process. We know that is emasculation. By emasculation, we can remove the anthers before the maturity of pollens or pollen grains to avoid self-fertilization. We can avoid self-fertilization by the process of emasculation by cutting the anther before the maturity of the pollen grains. Clear? So now we will go for the how the different characters are chosen, what are the sharp characters found in the pea plant, and how Gregor Johann Mendel continued his experiment. So remember, experiment on pea plant by Gregor Johann Mendel continued for about 7 years, between 1856 to 1863. Between 1856 to 63. For 7 years, the works of Gregor Johann Mendel continued. So he worked on the pea plant for all his experiments, and it took around 7 years. So between 1856 to 1863, he worked on Pygium sativum to give his different laws and prove the importance of factor. So now let's see what are the contrasting characters taken by Gregor Johann Mendel on a pea plant. So during the experiment of Gregor Johann Mendel, he took different characters of the pea plant, and they are particularly 7 characters are there which were taken by Gregor Johann Mendel. So number 1 is the stem height. First one, the stem height, which may be tall or dwarf. The character may be tall or dwarf. Likewise, the second one is the flower color. That is that the flower may be violet or white. Next, the third one is the flower position. That may be axial or terminal. Next one is the pod shape. Number 4 is the pod shape. The pod shape may be inflated or constricted. Next one, that is the pod color. It may be green or pod color may be green or yellow. Next we will go for the seed shape, which may be round or wrinkle. The sixth one is the seed shape, which may be round or wrinkle. And the seventh one is the seed color, that may be yellow or green. It is the seed color that may be yellow or green. Okay, so this is all about the characteristics taken by Gregor Johann Mendel, the contrasting characteristics. When Gregor Johann Mendel took the stem height, that may be tall or dwarf, the stem may be tall or may be dwarf, the flower color may be violet or white, flower position may be axial at the side or terminal at the top, pod shape may be inflated or constricted. Pod means the cover of the seed. The pea is present inside a cover which may be inflated or plain from upper side, from outer. If we look outside, it may be plain or may be constricted. The seed can be easily viewed from outside. How the seeds are present, their structure can be viewed easily in case of constricted pod. Next one is the pod color. The pod may be green in color or yellow. Sixth one is the seed shape, which may be round or wrinkle. Wrinkle means it is not rounded structure, but like this. Okay, so the seed color is the seventh character, which may be yellow or green. So first I have written the dominant character and then the recessive character. What is dominant and recessive, we are going to discuss about that. Okay, so these are the different characters, the 7 characters of the pea plant, which are taken by Gregor Johann Mendel during his 7 year of experiment between 56 to 63, 1856 to 63. So now let's see what Gregor Johann Mendel did during his experiment in detail. Okay. Now we will discuss about the experiment of Gregor Johann Mendel. So first let's discuss about the monohybrid cross. Monohybrid cross means it is a cross between by taking a single character. So mono means, we know the word mono means one, and by taking one character, Gregor Johann Mendel did his experiment monohybrid cross to find out the offsprings, the characters, how travels from the parental generation to their offsprings. So look at this. One character here is the length of stem. It is the stem length. So the plant may be, the stem of the plant may be long or short. When long, it is a tall plant. When short, it is a dwarf plant. Clear? So the character tall is represented by 2 alleles. Both are capital. And the 2 alleles representing dwarf plant is small t. Both are small. Clear? So when Gregor Johann Mendel crossed a male plant with a tall stem with the stigma or the pistil of the plant with dwarf stem or short stem, what was the result? Let's see. He took tall plant, I represent it with capital T, dwarf plant small t. These are parent 1. So the gametes from the father is T. This is the female plant, mother plant, and this is the father plant. The gametes are represented with G1. When you cross between these 2 plants, the result is capital T small t. This one is F1. F means first filial. Clear? So this is the result of Gregor Johann Mendel's monohybrid cross. In the F1 generation, he got a plant with characters of both the parent, male as well as female. But he found that phenotypically this plant is tall. This plant is tall. although there is a mixture of the character, the genes of the dwarf as well as tall plant, but the offspring is here tall, and this is not pure tall but hybrid tall. It is called hybrid tall, and this condition is called heterozygous. It is called heterozygous condition because both the alleles are different. One is capital T, another is small t. But here it is pure, this one is pure tall. The parents are pure tall and pure dwarf So these conditions are called homozygous condition. So heterozygous and homozygous, there are two conditions. When the alleles are similar, they exhibit a pure structure which is homozygous, and when the alleles are different, representing two different characters, it is called heterozygous condition. After getting the first filial, Gregor Johann Mendel did self-pollination with this plant. So here the F1 hybrid tall is self-pollinated, means the F1 is treated as parent 2 and it is self-pollinated. It is crossed with itself, means it is crossed with the same female, or the male plant is crossed with the same type of female plant, means the hybrid tall. Both are hybrid tall. So now the G2 will be either capital T or small t here in the first case, and here also either capital T or small t in the second case. When the capital T fuses with the capital T, it will get a homozygous tall plant, a pure tall plant. When the capital T fuses with the small t and the small t fuses with the capital T, in these two cases, it will give you hybrid tall plant. And when the small t fuses with the small t, it will give rise to a homozygous dwarf plant, which is pure dwarf. So these are the three varieties of plants we get. Gregor Johann Mendel got at the end of his experiment of monohybrid cross, where there are two types of plants. One is pure tall, two are hybrid tall, and one is pure dwarf. So the ratio, the genotypic ratio here in the Mendels monohybrid cross is 1:2:1. That is, 1 is pure tall, 2 hybrid talls, and 1 pure dwarf. So this is the genotypic ratio of Mendel's monohybrid cross. Here, so he got three different types of plants if we will study the gene, but physically, if we look from outside, the hybrid talls are equal as the pure talls. So if we will consider phenotypically, then the phenotypic ratio will be, these three are same, that is tall, and this one is dwarf, so it is 3:1. So 3:1 is the phenotypic ratio of Mendel's monohybrid cross, whereas 1:2:1 is the genotypic ratio. So how we get this? This one will be this one F1 G2, F1 becomes P2, so it is F2. In the offspring, we will get pure tall, hybrid tall, and pure dwarf types of plants. So we can show this through a square which is known as the Punnett square, and it is known on the name of Reginald C. Punnett, the scientist Reginald C. Punnett. So how we will show these things through Punnett square? Let's see. So if I will represent the first cross, this one, through Punnett square, what will be? It will be shown like this. It can be seen like this. Yes. Here we have capital T, 2 two types of alleles, and here the female plant is having small t, small t. When the capital T fuses with the small t, it will give rise to capital T, small t. Here, when the capital T fuses with the small t, it will also give capital T, small t. Here also it will be capital T, small t, and here also it will be capital T, small t. So this representation is known as Punnett square. And here all the offsprings are hybrid tull because the F1 is having hybrid tulls. So due to fusion of pure tull and pure dwarf, or homozygous tull and homozygous dwarf plant, we will get heterozygous tull plants. Likewise, if you will represent this cross in a Punnett square, we will have the structure like this. Here we have capital T, small t, and capital T, small t, because we are doing self-pollination here in this case. So the capital T, when fuses with the capital T, it will lead to homozygous tull. Capital T, small t will lead to heterozygous tull. Capital T, small t again leads to heterozygous tull, and small t fuses to form a homozygous dwarf plant. So this one we can write down capital T, small t, which may be female or male. Let's take it as female, this one as male. Clear? So this is representation of offsprings produced from parents through Punnett square. Clear? So let's use mathematics in genetics, in biology. So here we can write that the production, that is 1:2:1, the ratio we can get from by making a binomial expression. By making binomial expression, how can we get the thing here? Let's see. Look at this, the binomial expression ax plus by whole square. So here it is half capital T plus half small t whole square, which means half capital T plus half small t whole into half capital T plus half small t, and by multiplying this we will get half capital T into half capital T, that is 1/4 capital T, then plus half into half, 1/4 capital T, small t. Then by multiplying this again, we will have 1/4 capital T, small t, plus we will have half t and half t, 1/4 t square or t into t. So we have the result 1/4 capital T plus 4. By adding these two, we will get half capital T, small t plus 1/4 small t, which means at the end we will get 1 out of 4 as capital T means homozygous tull, half means 2 out of 4 we will get heterozygous tull means capital T, small t, and 1/4 we will get homozygous recessive, that is small t and small t. So this is the result of Mendels monohybrid cross, that is 1/4 are pure tull, half are hybrid tull, and 1/4 are pure dwarf. Another thing is also there in F2 generation, the phenotypic ratio is 3:1. That means 3:1. That means 3 of 4 are tall and 1 of 4 are dwarf. So this is the result of Mendels monohybrid cross, and from this he postulated two laws. He gave two laws that are very important. The first one is the law of dominant and recessiveness. That means the character which is found in the F1 generation, that is tall, that character is called dominant, and the character which is suppressed or masked, that is dwarf, is the recessive character. So the one which is seen is dominant, the one which is masked is recessive. Likewise, the second law is the law of segregation. So the law of segregation means the two alleles are present together in the parents, but they separate during the formation of gamete. So they segregate into two separate alleles and they migrate to their offsprings. So the traits, the alleles present together here but separate during formation of gamete and travel to their offspring to represent their specific character. So this is the law of segregation. Another thing we should notice here that at the P1 first, during the start of the process of monohybrid cross, Gregor Johann Mendel took two characters, one is pure tull and another was pure dwarf, and these two grandparental characters are found in their grandchildren. So here the characters of the grandparents are here, it is pure tull and it is pure dwarf, and the characters of their parents is also found here. So from this it is concluded that the characters from parents to offspring and from offspring from grandparents to their grandchildren, the characters continues to travel. Clear? So this is all about the experiments of Gregor Johann Mendel on Pygium sativum by taking a single character. This is all about monohybrid cross, law of dominant and recessiveness, and the law of segregation. Thank you very much.